Okay, hello. I'm Amanda Marsden, and this is my e-report, my first e-report for Cell Molec 1. This e-report will discuss various topics that revolve around DNA replication, such as chromatin compaction, DNA replication, DNA mismatch repair, the assembly and purpose of the pre-initiation complex, post-transcriptional mRNA processing, translation, combinatorial control, and the mediator complex, and RNAi. Chromatin compaction. So what are chromosomes? Chromosomes are long DNA segments that have been severely condensed, wrapped, and compacted. They are organized to be nicely contained within the cell's nucleus and to be easily dealt to daughter cells during cell division, known as mitosis. Each cell contains two almost identical copies of each chromosome, with the exception of gametes and specialized cells known as a homologous pair. One chromosome comes from the mother and one comes from the father in the case of humans. Males have 22 pairs and an XY pair, and females have 23 pairs of chromosomes, which means humans have in total 46 pairs of chromosomes. Each species has a different number of chromosomes, but that doesn't affect the organism in any way. Chromatin is DNA that is packaged along with proteins. So when we mention chromatin is the same thing as DNA, it's just that it's packaged and not just a simple double helix molecule. Chromosome compaction in bacteria cells is very different from human cells. Bacterial cells have circular chromosomes, and their genomes are packaged in a different way. It's done by supercoiling, which is when the DNA is just wrapped and twisted over and over and over and over and over again. It's kind of like a really, really tight wound up rubber band. And it's just placed inside the cell. The picture I have shows the difference between bacterial chromosomes and human chromosomes. So we obviously have a lot more DNA than bacterial cells, but just the difference on how it's compacted is shown. Everything else in this e-report is going to be about your eukaryotic cells. So this is just a little tidbit. So why compact chromosomes? Chromatin compaction is needed because each cell has around 6 feet of DNA. Now, we have billions of cells in our body, and if each cell has 6 feet of DNA, it needs to be completely and severely organized somehow. That DNA must be organized because it needs to be monitored and easily accessible for regulation purposes, which means that compaction must also be reversible. So, compaction is needed also for cell division, for example, mitosis. During interphase, DNA needs to be accessible for replication, but during the M phase or mitosis phase, chromosomes are found to be a lot more dense because that's when the chromosomes are separated for dividing. So they don't really need to be accessible at that point. So that just shows how sometimes chromosomes need to be more accessible than other times. So the photo I have just shows the process of the condensation of chromosomes. We're going to go into more detail further on in the report, but it shows how compaction starts, with DNA as a double helix molecule, and it just evolves as DNA just keeps compacting further and further and further into the regular chromosomes that we all know. So chromosomes contain long strings of genes, which are DNA sequences that can be transcribed into an RNA molecule. Protein coding genes are found on dense chromosomes because they have the most DNA space. Non-protein coding genes are found in inefficient genomes where large amounts of intergenic DNA is found. Intergenic DNA is DNA that does not code for proteins, which proteins are basically what we're all after. So intergenic DNA isn't really of use to us right now. The nucleus holds all the genes that are involved in making RNAs for the ribosomes that are needed for DNA replication. Okay, so one of the first levels of compaction is the formation of nucleosomes. Nucleosomes are the basic units of chromosomes and are made up of 4 different kinds of histone proteins. These different kinds of histone proteins are called H2A, H2B, H3, and H4. 2 copies of each histone protein comes together to form an octamer, which is shaped like a spool. Each of the 4 histones have a tail that protrudes out that is their protein. DNA wraps tightly around the histone spool, which around 147 base pairs is what wraps around the nucleosome. Sections of DNA that are devoid of proteins and are not coiled around the spool are called linker DNA. So the picture I have up in the corner just shows an example of just one nucleosome. So each of those little blue circles or balls is an example of a histone protein, and they're each labeled. And then the DNA, you can see the DNA is circled around it, and this little where the arrow points to linker DNA, that's where there's no proteins around DNA, so it's just open. So that's an example of the linker DNA. And here you can just get an overall idea of the spool shape of the nucleosome and how DNA is wrapped around in the middle. And this is also a good example of histone tails that protrude out. A lot of images don't have the histone tails, so in further slides, just know that the histone tails are there, you just might not be able to see them. So DNA binds to the histones non-specifically, which means DNA has an overall negative charge, and the histones have a positive charge, which attracts the DNA. As DNA is wrapped around the histone proteins, the original linear strand of DNA is shortened. And as the DNA exits one nucleosome, it is led to another, which forms a long chain of a bunch of nucleosomes, which kind of resembles beads on a string, which you can see the picture I have to the right. DNA is compacted to 1/3 its original length once it's wrapped around nucleosomes. DNA rarely exists in this form, and you can see that I attached another picture of different nucleosomes. This one is a little bit easier to see how the octamers are shaped because each histone protein is a different color. But obviously here you can't see those histone tails we were just talking about, but it's just another visualization. The next level of compaction after the nucleosomes are formed is what's called the 30 nanometer fiber. The 30 nanometer fiber is the least packaged form of DNA. It requires the histone 1 protein, which is a linker histone, found on the edge of the nucleosome where DNA exits the spool on its way to the next nucleosome. H1 changes the path of DNA and promotes more twisting. The 30 nanometer fiber is coiled to form the 10,000 times compacted mitotic chromosome, which is the normal chromosomes seen on an image earlier before. H1 coils nucleosomes into a tight helix or circular shape, which is seen in my photo to the right. So you see the nucleosomes in a different kind of fashion, and in the middle is the histone 1 proteins, which bind and cause the nucleosomes to kink and kind of fold up tighter. And those compress or condense very, very tightly to form the 30 nanometer fiber. On the next slide, there is a movie that shows, a little short video that shows the compaction of DNA. So if you feel free to pause the movie and watch it if you want. So this next video describes the whole process of chromatin compaction that we just discussed. So it starts at the DNA molecule as a double helix. It condenses down to a nucleosome. Once histone 1 proteins are added, it coils tighter into a 30 nanometer fiber. That 30 nanometer fiber is further compressed into a 250 nanometer fiber, and that compresses and twists more to form the mitotic chromosomes. So next we're going to talk about chromosome regulation. Cells need to have on-demand access to different DNA sequences. By regulating chromatin, cells can also regulate access to DNA. This means having the ability to unwrap specific sequences while keeping the rest of the chromosome tightly packaged. This is done by chromatin remodeling complexes. These complexes use energy to form ATP to move nucleosomes out of the way. These complexes physically force DNA to unwrap from one nucleosome while threading adjacent DNA onto the next one. These complexes free up short regions of DNA by making it accessible. Chromatin remodeling complexes can also rearrange clusters of nucleosomes by spacing them out, which is thus exposing more linker DNA. So this image shows what chromatin remodeling complexes can do. So this image shows different ways that chromatin remodeling complexes can open up DNA regions, but they still all processes when they use force to make these segments more available. Chromosome regulation can also be done in a way that involves the histone tails on the nucleosomes. These tails are rich in lysine and are positively charged. So one process is the use of histone acetyltransferases, or HATs. These require the use of ATP, and they acetylate the histones, which neutralizes the positive charge by adding an acetyl group. This weakens the bond with DNA because DNA is a negative charge. So this makes the nucleosome unstable and allows access to DNA because DNA can unwind from the nucleosome. The opposite of that is the use of histone deacetylation complexes, or HDACs. These deacetylate histones by removing the acetyl group, which restores the positive charge on the nucleosome and locks access to DNA, because since DNA is negative, it's more attracted to the positive charge, which keeps it closed and tightly bound. So the photo shows an example of what we were talking about. So the hats add those acetyl groups to the tails and makes the nucleosomes more spaced out with more linker DNA, and the HDACs remove the acetyl groups and make everything much more compact by restoring that positive charge. The histone code is a pattern of modifications that recruit or activate different enzymes at DNA. So they can call for transcription proteins, or they can send mitosis signals. There are different states of interphase chromosomes. Eurochromatin is accessible DNA, so it's less condensed, and it also usually has genes. And heterochromatin is more condensed, and it's more tightly compacted chromatin, and it is not as accessible. So the picture shows an example of this. The next section we're going to discuss is DNA replication. The overall structure of DNA allows replication to occur. DNA is a double-stranded and anti-parallel helical molecule. Base pairing enables DNA to be intrinsically replicatable. Using one strand of DNA as a template will create two new double strands of DNA that are mirror images of each other. DNA replication involves the copying of one molecule into two. The information needed for replication is intrinsically contained in the DNA molecule itself through base pairing. Adenine binds with thymine, and cytosine binds with guanine. Those four different molecules are the different bases that make up DNA. Each time DNA is replicated, a new molecule has one old strand and one new strand, which is known as the semi-conservative replication. So the picture to the right shows the semi-conservative model of DNA replication. The old strand that was used is in the darker blue color, and when it's replicated, the two new strands of DNA that are formed each have a blue and a lighter purple color. So that means that each of the original strands were used as a template to create the lighter purple strands. So that's semi-conservative replication. To separate DNA in vivo, the origin of replication is formed at specific DNA sequences. DNA helicases are bound to DNA, and they pry apart the double-stranded molecule by breaking the hydrogen bonds. Once separated, the single-stranded binding proteins keep the newly separated single-stranded DNAs separate from each other, so that they don't reattach. This creates what's known as a replication bubble. The site of DNA replication is basically started and maintained in this replication bubble. Humans have over 10,000 replication bubbles because DNA isn't just replicated at one spot at one time. It's happening many places through many cells. All of the replication machinery, or enzymes, are attracted to and binds to the replication bubbles. On each side of the replication bubble is a replication fork, which is a Y-shaped junction where double-stranded DNA is separated and new DNA strands are made using the old ones as templates. Each fork moves in opposite directions of each other, which means they're bidirectional. So the photos to the left show different origins of replications. So the first strand has two different ones, and next to the number 2, it shows how what started as a small origin of replication, how they're moving bidirectionally and are increasing in size, which means that the strands are separating apart with helicase and they're being replicated. So this shows how the origins of replications are in the replication forks are bidirectional. Hydrogen bonds are very weak, so that allows for little energy to be used when the bonds are being broken to separate the strands, which creates the origin of replication. So DNA can only be synthesized 5' to 3'. So for the example to the right, for the top strand, that's fine. So DNA synthesis travels towards the left. That's known as the leading strand, and it's made continuously because there's no issues there. But the bottom strand has to be made in segments because it's moving 3' to 5'. This is known as the lagging strand, and it is made discontinually. Each of those little segments, so notice how the arrows are in different, there's 3 different arrows because there's 3 different segments. Each of those segments is called an Okazaki fragment, and they're short segments of DNA that will eventually get joined together. So the bottom picture is just another example of the origin of replication, but it's closer up. So each of those smaller black boxes represents a replication fork, so that shows you how there's 2 on each side and DNA goes in opposite directions. Many different enzymes are required for DNA replication. DNA polymerase makes a new DNA strand using the old one as a template. It makes a phosphodiester bond between the 5' phosphate of an incoming nucleotide and the 3' hydroxyl on the end of a newly synthesized DNA strand. Individual nucleotides enter the reaction site of DNA polymerase as deoxynucleotide triphosphates, or DNTPs. So adenine, for example, would be DATP, or deoxyadenine triphosphate. They each carry the same amount of energy as an ATP molecule, and the energy needed to power DNA replication comes from the cleavage of the phosphate on the DNTPs. So one of the phosphates is removed. Clamp proteins are needed to keep DNA polymerase from floating away. So clamp proteins encircle DNA and interacts with DNA polymerase, which also requires a clamp loader protein to put the clamp loader onto DNA. This speeds up the rate of DNA polymerization, and for the lagging strand, it's added and removed several times. So this picture just shows some of the players we just talked about. You can see the leading strand and the lagging strand. You can see the sliding clamp, which is here. You can also see the separate strand binding proteins. You can see the DNA polymerase creating the new strands, adding the bases, and you can see helicase opening up DNA so that it can be replicated. Okay, so DNA polymerase can only link the 5' ends of new nucleotides to the 3' ends of nucleotides that are already there. So RNA polymerase is needed because it makes RNA strands that are complementary to DNA from scratch. An RNA primer is formed by primase that provides a free existing 3' end that DNA polymerase can use to begin adding new DNA nucleotides. The leading strand only needs one RNA primer at the very beginning because everything else is made continuously. But the lagging strand needs several because it's made discontinually. Each Akazawa fragment on the lagging strand is a new segment of DNA which needs its own primer. Combining Akazawa fragments together requires 3 different enzymes. There's one enzyme that lands on the 3' end of a fragment and chews up the RNA primer on the fragment in front of it. Repair polymerase puts down new DNA in place of the just removed DNA, and DNA ligase joins the phosphate sugar backbones of adjacent DNA fragments together by forming a phosphodiester bond. So this is just the picture previously shown, but now we know all the players in the picture. You can see the RNA polymerase at the bottom working together with primase, form an RNA primer. You can see DNA polymerase adding bases, and further down you can see ligase joining the 2 Akazawa fragments. We also see the single-stranded binding proteins on both the leading and the lagging strands that keep the 2 single-stranded strands separated. You see the clamp proteins next to the DNA polymerase on both strands, and this just displays a good overall view of the enzymes and what they do during replication. So next is a video that shows this in real motion. Feel free to pause and play it if you want to. Primase can only lay down RNA primers that are in front of itself. So this creates a problem because how can we remove RNA primers from the end of DNA where there's nowhere, for RNA polymerase to land? If left this way, then the chromosomes would shorten with every single round of replication and would eventually become nothing. So DNA caps were evolved called telomeres. They provide a landing pad on DNA strands for DNA replication machinery or RNA polymerase to attach. They are made by the enzyme telomerase, and it carries its own RNA template strand, which allows it to make short DNA sequences many times at chromosome ends. This forms a cap with many tandem repeats that cannot be shortened. Um, Excess strands are telomere segments, and it just codes a bunch of junk DNA that's not important. So this junk DNA is what's chewed into during the next replication cycle, which prevents the real important DNA from being chewed up. Mutations are permanent changes in DNA sequences that can drive evolution, mostly for negative reasons, but sometimes for positive. Um, these changes can be devastating to an organism if the mutation is in a vital region of DNA, which is a region that codes for protein or protein structure. There are several different kinds of mutations. One example is sickle cell anemia, which is a base change mutation, which is when an A is changed to a T, and that changes the whole structure of the red blood cell. DNA itself is self-correcting. DNA polymerase checks the nucleotides as they enter the reaction site and ensures that they are the right fit. DNA polymerase proofreads 3' to 5', and it's done by touch. So if the wrong base is attached, DNA will lose its smoothness, and the error is felt. If the error is felt, DNA polymerase shifts the offending nucleotide to a different domain of the enzyme called exonuclease, and here the phosphodiester bond is cut, the wrong nucleotide is clipped off, and once that happens, DNA re-enters the synthesis portion of the enzyme, and DNA polymerase fixes the replication error. There are different DNA repair mechanisms in addition to proofreading. DNA mismatch repair system is dedicated to fixing mistakes that sneak through the polymerase proofreading. It fixes bases that are not correctly paired, which is around 99% of mutations that occur. The error is recognized as a distortion of the DNA backbone, and a new section of newly made DNA with the mismatch is cut out. The old non-mutated strand is used as a template to fix the error. In bacterial cells, methylation is attached to the old strand that is targeted as good and faithful. And so in bacterial cells, they know to use the methylated strand when going to fix mutations such as these. In humans, we don't know how cells just know which strand is the trustworthy strand or not. It's still kind of unknown for us. But mismatch repair is the best defense against somatic mutations, which are mutations that occur in any cell that's not a gamete. Double-stranded break repairs is when a complete DNA molecule is severed into two pieces. So that's seen by the two circles on the picture to the left. So there's two breaks, one on each side of the molecule, so the whole piece is severed. Because both strands are cut, there is no old strand to use as a template for repair. This can be caused by different reasons, such as ionizing radiation, or mistakes that occur at the replication fork. There are two ways to repair double-stranded breaks. One is homologous recombination, and one is non-homologous recombination. Non-homologous recombination is when a nuclease is used to file down the two ends of DNA, creating blunt ends. DNA ligase is then used to fuse the blunt ends together, which causes a deletion of nucleotides at the repair site. When the deletion occurs in intergenic regions, it's okay because those are no protein coding areas. But when the deletion occurs in the middle of a gene, it's almost rendered inactive because it's rendered as a loss of function mutation. Non-homologous recombination is viewed as a last chance fix, which is either like do this or die. And it's always available, but it's not always the best choice. Non-homologous recombination is preferred because it's a cleaner fix. It requires a nearby whole DNA molecule that is similar in sequence to the broken one. This serves as the repair template. Everyone has two complete DNA molecules that are almost identical, which are the homologous chromosomes that you get from your mom and dad. They're not necessarily located to each other, which is the part that makes this repair kind of tricky. Homologous recombination is the most versatile and efficient repair, but like I said earlier, it can be very hard to find a similar DNA molecule. The process of homologous recombination is first done by using nuclease that chews back one of the two strands of DNA at the double-stranded break. This frees single-stranded broken stretch of DNA samples nearby DNA molecules for similarity in sequence. If a similar sequence is found, strand invasion occurs, which is where a single-stranded broken molecule displaces one of the two strands of the intact molecules and base pairs with it, creating a branch point. The branch point is where two DNA strands cross, one from each molecule. Invading strand is then elongated by a repair polymerase, and as DNA is synthesized, the branch point moves forward by helicase to allow for additional DNA to be made. The invasion strands are cut to normal length and then sealed with DNA ligase, which results in double-stranded break being corrected. So this is shown in the next picture, and this is also done during phase 1 of mitosis, which is when crossing over occurs, which is the switching of chromosomes to increase diversity. So next we're going to talk about the assembly and purpose of the pre-initiation complex, which is part of transcription. Transcription is the process of copying a particular region of DNA into RNA so that it can be used for protein synthesis. Each gene contains the instructions for building proteins. Different proteins are needed for different cellular processes. So for transcription to occur, several enzymes are needed. These include different kinds of RNA polymerase 1, 2, and 3. Additional protein complexes are needed because RNA polymerase can't initiate transcription by itself. So general transcription factors, or GTFs, are assembled at the promoter along with RNA polymerase 2, which altogether forms a pre-initiation complex, which allows transcription to proceed. The pre-initiation complex is formed of the GTFs at the promoter site with RNA polymerase 2. GTFs are responsible for recruiting the RNA polymerase to the promoter. They position it, they separate the double-stranded DNA of the gene that is targeted, and launch RNA polymerase down the targeted gene. The first GTF to recognize and bind to the promoter is TF2D, or transcription factor D that calls RNA polymerase 2. This GTF, a subunit is included that is called TBP, or the TATA binding protein. TDB binds to the promoter sequence called a TATA box, which is rich in T's and A's found upstream from the promoter, and it's rich in thymines and adenines, which are the T's and A's, because they only have two hydrogen bonds, which are easier to break than cytosine and guanines. When the TBP binds, it kinks the DNA to a 90-degree angle, which makes the Tata box promoter unmistakable for other GTFs so that they can all assemble together. The order of GTF assembly is transcription factor 2D, transcription factor 2A, B, RNA polymerase, transcription factor F, transcription factor 2E, and H. Next is a picture that shows this. It can be kind of confusing, so mainly focus on the black boxes that are highlighted. So A shows transcription factor 2D that is paired with the Tata box binding protein, and that lands on the Tata promoter sequence. So B shows that once that's bound, the other transcription factors start flowing in, which also leads to RNA polymerase coming in and all the other transcription factors, which allow RNA polymerase to eventually let loose and flow down the gene and start transcribing the DNA. Post-transcriptional mRNA processing is a different process that is done to mRNA once it's done to tell the cell that mRNA is completely made so it can leave the nucleus. The mRNA must leave the nucleus to reach the ribosome in order to get translated into proteins. So different mRNA processing includes capping, polyadenylation, and splicing. Enzymes that are needed travel along with RNA polymerase during transcription. Post-transcriptional mRNA processing allows for quality control because this way the cells can inspect the mRNA for each of these different targets before it is translated outside of the cell. So the first process is capping. Capping is when the mRNAs receive a methylated guanine base that is attached to the 5' end of the mRNA, which marks the beginning of mRNA as genuine. Adenylation is the addition of a poly-A tail, which is a run of adenine bases. This is added to the 3' end of mRNA, which marks the mRNA as genuine. Both of these processes add stability to mRNA and labels the mRNA as legitimate mRNA to the rest of the cell. Proteins are recruited to both of these areas, which cause mRNA to circular size. PABP is the poly-A tail binding protein, which is attached to the 3' region of mRNA, which attaches to the poly-A tail, and the cap binding protein is attached to the cap of mRNA, and together they bind, allowing mRNA to circular size. Splicing is also done because mRNA is made out of sections of sequences called introns and exons. Introns are non-coding sequences of mRNA, and exons are coding sequences of mRNA. Both are transcribed because RNA polymerase can't discriminate between coding and non-coding genes, so splicing is the process of removing introns and stitching together exons. Each intron has conserved sequences at their ends and the middle, which serve as codes for removal. Spliceosomes are used to remove introns and ligates exons together. The freed 5' end attaches to the middle of the intron, making a lariat. Alternative splicing can also be used in this same kind of concept because not all exons must be kept. So by splicing non-adjacent exons together and deleting others, new proteins can be made from one gene, which allows one gene to be used in multiple ways. So the picture shows the process of splicing, so that's removing the intron regions and ligating together the exon regions to form the protein coding sequence, which is then translated. So when mRNA is finalized, it becomes circular, like I said before, due to the different proteins. And this is the only way that mRNA is allowed out of the cell, which is a regulation factor. The photo on the right actually shows translation, but I think it shows good detail of the poly A tail and the 5' cap and how those two bind together to make mRNA circular. Our next topic is going to be translation, which is the process of DNA and RNA nucleotide sequences being translated into amino acids, which produce proteins. mRNA sequences are translated in sets of 3 nucleotides known as codons, and they go by a genetic code, which is an amino acid dictionary that shows what each group of bases codes for. Each codon codes for 1 amino acid because there are 20 amino acids and only 4 nucleotides. So in order for each amino acid to be coded properly, there needed to be 3 bases. For any sequence of RNA or DNA, there are 3 different reading frames or ways to read codons. Only one is correct, and that is an open reading frame that starts with a start codon, which is AUG, and ends with a stop codon or a termination codon. Translation uses tRNAs, which is an adapter molecule that matches amino acids to mRNA codons. Codons and mRNA do not directly bind to their corresponding proteins. tRNAs are adapter molecules that bridge the two. This is done using an anticodon, which reacts and binds to mRNA. This directly base pairs with the codon, and it must be 3' to 5' because it has to be antiparallel to the mRNA. The anticodon contains a corresponding amino acid on the other end of the tRNA. tRNAs have intramolecular base pairing. tRNA is T-shaped with 4 regions, and the amino acid would be attached to the pink region at the top. There are not enough different tRNAs for there to be 1 tRNA for each codon, so this introduces wobbling base pairs. Wobbling base pairs allow 1 tRNA to recognize multiple codons. This usually occurs at the 3rd position of the codon. There are 31 different tRNAs to accommodate the 61 different amino acids, so some are specific, but most wobble to make up for the vast difference. So how do amino acids attach to each tRNA? Well, tRNA charging is done by aminoacyl tRNA synthases that are responsible for charging tRNAs with amino acids. There's usually 1 enzyme for each amino acid, and tRNAs differ by the specific sequences of nucleotides in the anticodon region as well as the amino acid attachment point. So these different processes make translation possible. Ribosomes are very big players during translation. They are huge pieces of RNA and protein molecules that come together to form a complex. They have one job, which is protein synthesis, and they are made of 2 subunits, a large subunit which forms the peptide bonds between amino acid, and a small subunit that matches mRNA codons to their proper tRNA. The 2 subunits sandwich mRNA, and initiation ensures proper placement of the 2 different subunits. The larger subunit has 3 voids, which is shown on the picture on the right, which has an A site, a P site, and an E site. The A site is the acetyl site, which carries single amino acids. The P site, or the peptidyl site, carries peptide chains of amino acids, and the E site is the exit site. Translation is initiated by EIFs, or eukaryotic initiation factors, that bind to the 5' cap of mRNA in the small ribosomal unit. EIFs call all the enzymes needed for translation. An initiator tRNA is used that has methionine attached where other amino acids would be attached, and has the anticodon for AUG, which is the start codon. This tRNA associates with the small ribosomal subunit and keeps mRNA engaged while the initiator tRNA scans for an AUG start codon. When the start codon is found, the initiator tRNA base pairs to it. Finding the start codon establishes the correct reading frame. Without it, the wrong sequence would be read, and the resulting protein wouldn't be correct. The large ribosomal subunit joins the complex and associates with the small ribosome. When this happens, the initiator tRNA is placed in the P site of the ribosome. So the picture below shows the initiation of translation. It shows the initiator tRNA with methionine binding with the AUG start codon that was read on the mRNA, and then it also shows the larger subunit binding with the smaller subunit, placing that initiator tRNA into the P site. Once translation is initiated, another tRNA enters the A site and binds to the codon via base pairing. The amino acid on the tRNA that's in the A site forms a peptide bond with the amino acid chain that is linked to the tRNA on the P site. This transfers the entire amino acid to the tRNA in the P site. Translocation then occurs, which is where the entire ribosome shifts forward by one codon. The tRNAs stay base paired with their codons and don't move. The tRNA formerly in the P site is now in the E site, and the tRNA that was in the A site is now in the P site, with the amino acid chain on the P site as well. After this happens, the A site is now empty. When another tRNA enters the empty A site, the tRNA in the E site is emptied as well. mRNA is pulled through the ribosome until a stop codon is reached, which replicates this process over and over and over again. Only 2 RNAs can be in a ribosome at one time, which is why when one tRNA enters the A site, one enters the E site at the same time. So this photo demonstrates that fairly well. It shows the P site having the tRNA with the peptide chain, and then it shows a tRNA coming into the A site. It shows the transfer of that peptide bond, and then it shows how the peptide chain is transferred to the tRNA in the A site, but because it has that chain of peptides, it needs to be moved to the P site, which transfers all the other tRNAs, and it's just a cycle until a stop codon is reached. So the termination of translation is signaled by stop codons and release factors. Stop codon is positioned under the A site, and release factors enter the A site and bind to it, which stops translation because no tRNAs can recognize the stop codons because there are no anticodons. Release factors release the polypeptide amino acid chain from the tRNA that's in the P site, and that is free from the ribosome, and it's free to go off and form its own protein. So the 2 ribosomal subunits dissociate, and mRNA is released. So the picture below shows the whole process. It begins, with initiation, and it has the mRNA with the start and stop codons, the initiator tRNA, and the 2 ribosomes. It puts the initiator tRNA into the P site. It shows replication in the whole process. It shows elongation and the process of transferring that peptide bond. And then termination is seen with that stop codon and the release factor that's shaped differently and has no anticodon. So nothing else can bind to the A site, and release factors release that peptide chain, and so the peptide chain is left to circle off. All the segments and proteins and enzymes dissociate, and then the cycle can start all over again. Polysomes are mRNA with multiple ribosomes on it that are translating constantly. So this is like that picture that I showed you before, and it has a bunch of different ribosomes on it that are at different stages of translation. mRNA is being translated by multiple ribosomes at different points. New ribosomes assemble at the 5' end as soon as the processing ribosomes leave. So this just means we're constantly making proteins, and it's not just done one at a time. In this section, we're going to talk about combinatorial control. Groups of regulatory proteins, called activators and repressors, work together to precisely control gene expression. Activators facilitate transcription by allowing RNA polymerase to stably bind to promoters, which turns genes on and allows them to be copied. Repressors inhibit transcription because they prevent RNA polymerase from binding, which prevents genes from being transcribed, which turns them off. Combinatorial control is when there are many signals that are able to be expressed to regulate one gene. In this case, each regulator is sending its own signal that is received by a mediator complex that makes a committee decision on whether to express or repress a gene. Mediator complexes sit on the end of RNA polymerase II and relay information heard from signals from activators and pass them on to RNA polymerase II. The mediator complex receives all of the signals affecting a single gene and integrates them, just basically saying if they're like positive signals or negative signals. After combining the signals, the mediator sends the final result to RNA polymerase II, which sends activating or repressing signals that can receive signals from regulators at enhancers. So this is basically a form of gene regulation, and it uses different signals from like the environment or from our body to express or repress genes, and it's all done by like a judge and jury kind of thing. So the jury is all these different factors, and they form an opinion, and the judge, which is the mediator complex, thinks about their opinion, looks at it, and is the action person. He decides what's going to happen, which in turn turns the genes on or off. Our next topic is RNA interference. RNA interference, known as RNAi, is a mechanism that is used for gene silencing. This requires the information of double-stranded RNA. Once double-stranded RNA is recognized by RNAi, all double-stranded RNA and single-stranded RNA with the same sequence is destroyed. RNAi is believed to be a defense the body created to act against viruses, because viruses are the natural source for double-stranded RNA. RNAi is triggered by double-stranded RNA that is fully base paired and greater than 23 base pairs long. The longer double-stranded RNA molecules are cut by an enzyme called dicer. Smaller double-stranded RNA fragments are called siRNA, or small interfering RNA. Small interfering RNA is bound to an RNA-inducing silencing complex known as RISC. RISC uses one of the two strands from the siRNA that it's bound to recognize other RNA molecules with the complementary sequence and finds, and basically it destroys double-stranded RNA or single-stranded RNA. And RNAi is the most potent form of regulation. RNAi is amplified by RNA-dependent RNA polymerase, or RDRP. After RISC cuts an mRNA, two unstable fragments are left. RDRP uses one of the fragments as a template to generate double-stranded RNA, and it doesn't need a primer because it's RNA. Double-stranded RNA is cut by dicer, and the cycle continues. So the picture shows just the process of RNAi. So the microRNA precursor folds back in itself, and it's held together by hydrogen bonds. And an enzyme called dicer moves along the double-stranded RNA, cutting it into shorter segments. One strand of each of the shorter segments is complexed with RISC, and then RISC uses that target RNA that it's bound to find other sequences that are either double or single-stranded, to find and delete those so they're not circulating around the body. RNAi is another form of gene regulation and control. So that's all. Thank you for listening.